Dear students, Assalamu alaikum. Today I am going to deliver a lesson for class 12th, subject zoology. Our today's lesson is related to your chapter biotechnology. And in today's lesson we will discuss about the most important topic, the recombinant DNA technology. Now you have already studied about genetic engineering in our previous topic, which is alternatively called as a recombinant DNA technology or gene cloning. Paul Berg is considered as the father of genetic engineering, and Stanley Cohen and Herbert Boyer constructed the first recombinant DNA in 1972. Basically, genetic engineering is the manipulation of prokaryotic as well as eukaryotic DNA. It involves cleavage, that is the breaking of a DNA molecule at a desired place to isolate a specific DNA segment with desired genetic information, and then insertion of the cut segment in another DNA molecule at a desired position. The product thus obtained is called recombinant DNA, and the technology is called as genetic engineering or recombinant DNA technology. As I already told you that the first recombinant DNA was constructed by Stanley Cohen and Herbert Boyer in 1972, and in fact they cut the piece of a DNA as is depicted in this sketch. A piece of DNA was cut from a plasmid that was carrying antibiotic resistance gene. It was in the bacteria Salmonella typhimurium. Now, cutting of a piece of DNA from plasmid was with the help of restriction enzymes known as restriction endonuclease, also called as molecular seizure. The piece of DNA cut from the plasmid was linked with the plasmid DNA acting as vector. Now, linking of the piece of foreign DNA with the vector was done with the help of an enzyme called as DNA ligase, which acts on the cut DNA molecules and joins their ends. Thus, the newly formed DNA having these integrated fragment of antibiotic resistance gene is called as recombinant DNA. The vector is used to transfer the recombinant DNA to E. coli. When this recombinant DNA is transferred to E. coli, Escherichia coli, which is a bacteria, then what happens? It replicates in the new host in the presence of DNA polymerase, another enzyme, and thus makes multiple copies of recombinant DNA. This ability to multiply copies of antibiotic resistance gene in E. coli was termed as cloning of the antibiotic resistance gene in E. coli. Thus we see there are three basic steps involved in the process of this creating these genetically modified organisms, or we say this transgenic organisms. These steps include: number first, the identification of the DNA with the desired gene. The second step is the introduction of the identified DNA into the host. And thirdly, the maintenance of introduced DNA in host and transfer of DNA to its progeny. Now, this is all about the recombinant DNA technology. Now, in our next topic, we will discuss about the various tools of this recombinant DNA technology. Until then, goodbye.